hormone, you can probably deduce the function, the binding, the half-life, just very many characteristics of the hormone that you need to know as a PA. One of the important considerations with respect to the structure of the hormone is how it binds, where it binds, because where it binds will in large part determine its mechanism of action. We said that in general, and with the exceptions to all these general rules, but in general, if a hormone binds intracellularly, its primary function is as a transcription factor. And we defined a transcription factor as really anything, any molecule substance that modifies the copying of DNA to RNA. And so that's the primary function of these hormones that bind intracellularly. And hormones that bind at the cell membrane may or may not act as transcription factors, we're going to find out. But if they do, they have a much less direct, they must utilize a much less direct pathway to do that.
Many cell signals are small, lipid-soluble molecules that can readily cross the cell membrane, where they interact with a receptor protein in the cytoplasm or in the nucleus.
So once again, if the hormone can pass through the membrane, it can bind to a receptor, which is a protein. These proteins are going to start, these various components are going to start popping up fast and furious now. So that protein receptor can be in the cytoplasm or in the nucleus of the cell. And when the hormone binds the receptor, you now have a hormone receptor complex. Now the ambiguity here that you're going to encounter in your clinical reading is that you will read in various sources that most transcription factors are proteins. But understand that when we're talking about, because I'm arguing now that, for example, steroid hormones can be transcription factors. Really what they're doing is binding to an intracellular protein, and it's then that complex or the activation of that protein by the steroid that's functioning as the transcription factor.
Intracellular receptors may trigger a variety of responses. For example, some act as regulators of gene transcription. These receptors have a binding site for DNA. In the inactive state, the receptor cannot bind to DNA because an inhibitor protein occupies the binding site. When a signal molecule binds to another site on the receptor, the inhibitor is released and the DNA binding site is exposed. The receptor then enters the nucleus and binds to a specific nucleotide sequence on the DNA, which activates transcription of a particular gene, usually located adjacent to the regulatory site. The target cell's response depends on the nature of the cell.
So another key point here, which is sometimes overlooked when we celebrate the complexity of all this, make sure you appreciate that these hormones, more specifically the hormones and the receptor complexes together, must bind to the DNA. They bind to the DNA directly. Now, we're oversimplifying this somewhat, but when we talk about a transcription factor, it is an effect. It is an alteration of the ability of the polymerase to transcribe the DNA. And so for that to happen, there must be some direct interaction between the hormone receptor complex and the DNA. It's usually upstream. By upstream, we mean that there's some modification, some alteration of a promoter or some other regulator prior to the gene. But nevertheless, what this steroid hormone is doing, if indirectly, is altering the ability of that DNA to be copied in a manner that it otherwise would or would not be. Transcription factor. All right, so the animation referred to, for the most part, steroid and thyroid hormones. All of these, for our purposes, well, most all of them are synthesized from cholesterol, and that would include the sex hormones, the androgens, the estrogens we'll be talking about later, the progesterones we'll be talking about later. Also include the glucocorticoids. These are an important class of adrenocortical hormone that are important, as the name suggests, in regulating blood sugar levels, but they perform a variety of functions. They perform an important role in your ability to respond to stress, for example, and regulate metabolism. So they're very important in a variety of ways. The mineralocorticoids, we won't have much to say about in this discussion, but later on we'll find that they're very important in your ability to retain water, retain the water that you take into your body, so you're not diluting your cells or concentrating the cells inappropriately. And then vitamin D, which is otherwise called calcitriol, or goes by a long fancy name, is also classified as a steroid, although some of these have their own more specialized subclasses in biochemical classification systems. Now, the little table down here just points out that these steroid hormones can be synthesized in the adrenal cortex, and I would argue, as a PA, it's this first and third group that you want to focus on, because what we'll find, and as we look at the structures up here, what I want you to make sure you understand, and I will probably repeat ad nauseum, as I tend to do, is the same steroid, I guess the proper term is the same steroidogenic pathway. The same steroidogenic pathway is present in the adrenal cortex and in the gonads. And so it's simple, subtle regulation that changes which hormones are produced and how the relative concentration of hormones is adjusted. So keep that in mind. That will be informing the discussion for the remainder of our course. And then sometimes steroids can be generated peripherally. For example, the liver cranks out quite a number of both enzymes, proteins, regulatory proteins, and steroids. We've indicated that adipose tissue is a very potent, we're learning, a very potent endocrine tissue, and that means it not only cranks out hormones, chiefly hydrophobic hormones, but it also has receptors for such hormones. And so it's also regulated by the endocrine activity of these hormones. With respect to the transport of steroids, I think you understand the theme we talked about before. In general, if the hormone is relatively insoluble in the blood, that means it's going to need some help traveling in the bloodstream. Consequently, all steroids, to one degree or another, are bound to protein chaperones or carriers. And we'll talk more about the importance of the carrier concentration later. The problem with that, as we discussed before, is if all of these hormones have to bind to receptors, then the transporter can be a bit of an encumbrance. And that is to say, in most cases, not all, but in most cases, the chaperone must be detached before the hormone can then bind the receptor. So in effect, that prolongs, that's an additional step in the metabolism of this hormone, and therefore it tends to extend the half-life. In fact, we'll see that this is a trick that is used by some water-soluble hormones. They will bind to carriers to extend their half-life. Remember, most of these hormones have relatively short half-lives, with the exception of the hydrophobic hormones. So let's see, what have we not talked about? I think the main point that I've been trying to stress, both in that animation and in our introductory discussion of steroids, is that in order for the steroid to act as a transcription factor, it must bind intracellularly, either in the animation you saw that it bound in the cytoplasm and then the HR complex went into the nucleus, or it can bind directly in the nucleus. Now, if you look at the illustration here, please be sure that you understand, in order for this whole process to work properly, there must be a specific binding site on the DNA for that hormone receptor complex. It goes by a variety of names. We'll just call it a HRE, a hormone response element. So this is on the DNA. And so in this illustration down here, you can see the hormone response element is sort of a lighter color. It is also the case that steroids frequently bind the HREs cooperatively. And by that we mean that in order for the steroid hormone to function efficiently or completely as a transcription factor, it needs a little help. It may require binding of another hormone. It may be, and this is a little bit tricky here because it varies, it's just a wide spectrum of variation. It may require binding of two hormones together. That's usually called dimerization. We'll talk about dimerization in another context later, but you can see we have a dimer just means two of them paired together. On the HRE, sometimes they're two different hormones, and sometimes it's a hormone and some other molecule that acts to facilitate, again, sort of regulate the regulator. So just be aware that steroid hormones, when they act as transcription factors, often do so in a fashion like this, where there's sort of a cooperation between two receptors involving two of the same hormones, two different hormones, or a hormone and some other regulator. So we talked about what a transcription factor is. We've indicated that transcription factors often have a genomic effect. By genomic effect, we simply mean that, I think you know what I mean there, that somehow we're altering the expression of the DNA. So that's just another way of expressing really the same thing. I think I mentioned on another occasion that as the technology, as the science stands at the moment, we have a much more thorough understanding of steroid hormone receptors than we do steroid hormones. In many cases, we have isolated and identified receptors. In some cases, we even know kind of what they do, but we just haven't figured out what's binding to them and how they're actually regulating transcription. There's reference here to two classes or families of receptor, the steroid and the non-steroid. I'm not too concerned about that. Rather, what I want to suggest here is that we're going to treat all of the sterols and steroids more or less the same. If they're derived from cholesterol, we're going to pretty much argue that they have a similar modus operandi. They pretty much operate the same way. But keep in mind, again, this is just adding some fancy language to what we've already talked about. Nuclear hormone receptors, i.e., for the most part, steroid and thyroid hormones, have a ligand binding domain. What does that mean? Steroid hormone receptors have a ligand binding domain. What does that mean? The hormone is a steroid. What's it going to bind to? The receptor. So it's going to refer to that because we're talking about receptors here. So the receptor is going to bind to the hormone, and it's going to have a DNA binding domain. So that just sort of drives home that both have to occur. There has to be formation of the hormone receptor complex and binding to the DNA. And we've already talked about the HRE. We've simplified that too much. But that HRE is generally just a regulatory sequence upstream of the gene that is being regulated. Not always. Sometimes these regulatory regions are just light years seemingly away from in fact, they can be on a different chromosome. But in most cases, they're very near the gene to be regulated. So if you look at the example, this is just at the top left here, we have another example of this dimerization process whereby you have, in this case, thyroid hormone. We're going to be talking about thyroid hormone. This is T3 binding at a dimerized HRE, the hormone response element, and the regulator is something called retinoic acid. So you get a wide variety of regulatory dimers in these steroid hormones. If you look over here at the illustration on the right, once again, we have thyroid hormone. This time it's T4. We'll sort all that out later. But notice T4 because it's just so many objections to the rules we're going to make. But in any case, thyroid hormone is generally insoluble in the blood, so it's going to have a carrier. Carrier is something called thyroid binding globulin. So you can see the thyroid binding globulin sort of detaches its payload here. The T4 passes into the cell where it binds an intracellular chaperone. We don't need to worry about that too much now, but it's sort of a little fellow in there that greets it and tells it, welcome, where would you like to stay tonight? And then they kind of figure out that the thyroid hormone is going to end up in the nucleus where it's going to do some business with the DNA. And so there's going to be detachment of the intracellular chaperone from the thyroid hormone. But in the final analysis, this is what I want you to pay attention to, that thyroid hormone is going to bind to the DNA to, in some way, modify transcription. So we're going to find thyroid hormones also act as transcription factors. But this sort of summarizes the typical sequence of events that you would expect for a hormone that alters transcription.
How fast are we talking? Does that
Good
occur?
question. Bartek is asking about how fast this occurs. And you remember on an earlier slide, I had reference to sort of slow and fast. This process itself, what we're looking at here, can occur fairly rapidly. Now remember, we have to sort of dissociate a lot of things here. We know that these hydrophobic hormones, first of all, have relatively long, as hormones go, they have long half-lives. So they're going to be in the blood for a relatively long period of time relative to proteins and amines. The process that we're looking at here is relatively short. This whole thing can happen in less than a second. But what is the problem? Where's the slowdown? Why do we say that hydrophobic hormones have relatively slow mechanisms of action? Well, again, you're right. I agree with you there, and that's going to be a bit of an annoyance. But this whole process up to the DNA doesn't take very long. What is happening at that DNA? What William's saying, we have to make the regulatory RNA, and then what's going to happen then? We got to make proteins. That doesn't happen overnight. Literally, I guess it does. But it takes a while to crank out those proteins in sufficient concentration, and a lot of those are regulatory. A lot of those are going to be regulatory proteins. They may be enzymes, they may be cofactors. They have to diffuse to the proper location. They have to be packaged and so on, and they're going to be water soluble. We're going to get them into the blood through the membrane. That's where the slowdown is. Anytime you're talking about a transcription factor, to crank out all those proteins, or even if it's only RNA, doing some sort of RNA thing, like interfering with one reaction or another, to get all of that settled out and at the right concentration takes much longer. So that's why we say that these transcription factors have slow mechanisms, because they're fooling around with the DNA.
So it's slow even though it lasts about a second?
I'm sorry?
You're saying it's slow even though it lasts about a second?
No. What I'm saying is, from this to that can happen in a second. But that's immaterial. I mean, this happens all the time. What's important is what's going on with the DNA and what follows that you learned about in your basic biology and chemistry. I was just talking with somebody in the office the other day that I was looking at the international standards for anatomy and physiology courses, what you all are supposed to learn. And we don't get about a third of it in here. Why don't we get about a third of it in here? Because it's all cell biology that we assume you as an entering PA student have already had, and we don't have time to cover it. But we didn't talk about this. You should be familiar with transcription, translation, genetics, epigenetics, and all that. If not, you'll have a course coming up shortly. I'll help you with that. But that's the slowdown. This is the slowdown here.
Steroid hormones are not water soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
Which reminds me, I was going to bring in a creature. I'll have to do that next week, that I found in the backyard and have you identify it for me, but it looks a lot like that. All right. Now I've broken my computer. There we go. So, once again, just be sure you understand the derivation of these steroids. They're all derived from cholesterol. They all have that multicyclic string, that ring structure there. Might want to give that one a try.
No.
Kind of like a Stroop's test, isn't it?
Like, well,
I feel as if you're all just going into a slow coma. But you can see the relative similarity of these various steroid structures. And of course, once again, the adrenal cortex has the requisite enzymes to undertake these steroidogenic reactions. And the other location for steroidogenesis is in the gonads. And we'll talk about the variabilities there. But in many cases, there's a good deal of overlap, and it has developmental consequences for you as a PA. All right, so much for the steroids. We'll come back and talk more about these later, but let's move on to the peptides. We know that peptides are by and large hydrophilic. They tend to be relative, at least as hormones go, at least as these group of peptides that act as hormones, for the most part, they're relatively small, on the order of several to several hundred amino acids. Again, that's relatively small. So small, in fact, that some of them show up in the urine, and you're able to measure the circulating level, or at least deduce the circulating level based upon concentrations in the urine. That is to say, they get filtered right through. We'll talk about renal function later. For the most part, they circulate unbound. There are some exceptions. We'll talk about the growth hormone and its little brother there, the IGF. And remember, what some water-soluble hormones do is use chaperones, use carriers to extend their half-life. And otherwise, they're relatively short-lived, short-lived. These have to be packaged into secretory vesicles. And what we're looking up there, looking at the top here, is something that we've seen before. We're looking at the general sequence of events involved in the synthesis of a protein or peptide hormone. So back here, of course, is the ER, and there'd be the Golgi there. And so what you'll notice is that as this hormone peptide is gradually formed, it starts out in the ER, and then it's shipped in a vesicle to the Golgi apparatus. And then finally, if it's a hormone that's going on, if it's a peptide that's going to be living its life as a hormone, it's going to be packaged into a secretory vesicle and then exocytosed out of the cell. But let's talk a little bit more about the specifics of this. One of the interesting things of the way that these hydrophilic peptides are synthesized is that the factories, the area of the ER where this process begins is in the membrane. I think you probably remember granular and agranular, or rough ER and all that. You remember? So they begin in what are called embedded ribosomes in the ER, but it's still largely a part of the membrane, and as such, as it turns out biosynthetically, it is advantageous for this long, very, very long precursor hormone, precursor peptide, to have included within it a hydrophobic sequence of amino acids. So what we're saying is a peptide hormone begins life as a much larger protein or peptide that includes a hydrophobic sequence that is sometimes called the signal sequence, sometimes called the hydrophobic tail, goes by a variety of names. And in that form, and in that precursor form with the hydrophobic sequence, it's called a pre-pro hormone. Isn't it like a pre-pro hormone? At at some point, when the relative primary structure of the pre-pro-hormone is completed, it's ready for shipping. And by the way, this can happen in a variety of ways. I'm simplifying this. This process can happen in the ER or on the way to the ER. There's no consistent pattern here. But at some point, either prior to or during the transit of that more or less completed, at least in terms of primary structure, pre-pro-hormone, the tail sequence is no longer needed. The signal sequence is no longer needed. Signal sequence, signal peptide, it's chopped off. It's cleaved by an enzyme. When that happens, the pre-pro-hormone now becomes a pro-hormone. Right? So the pro-hormone is identical to the pre-pro-hormone, except that it has lost its hydrophobic signal sequence. Now, the pro-hormone, now in the Golgi, is going to be further modified. You probably remember from your basic biology or basic science courses something called post-translational processing. We finished the process of translation in the ribosome. We're now going to chemically modify that protein to suit its peptide, to suit it more for its position in life. And it's going to have a certain one. It's going to have one job. It needs a set of tools to do those things. If it's going to have another job, it needs another set of tools. So it's chemically modified. It could be methylated, glycosylated, phosphorylated, any number of things could happen to it. And that happens in the Golgi, all right, part of post-translational modification. And that's happening to the pro-hormone, all right? When that process is finished, and this, again, is a little tricky, don't always expect for consistency here, either the pro-hormone or the hormone, which would be a fragment, a cleaved fragment of the pro-hormone, is released into the secretory vesicle for exocytosis from the cell. So what leaves the cell as a result of all this may be the contents of, from a vesicle that contains pro-hormones or active hormones, or both. You'll see, in fact, insulin, for example, is secreted in both precursor and active form. And then you can see the product being liberated eventually into the capillaries and traveling through the blood as hormones are wont to do. But do we understand the distinction between pre-pro-hormone, pro-hormone, and hormone? Semantically, the difference between the functional difference between a pro-hormone and a hormone is that a hormone is biologically active. In most cases, pro-hormones are not, or if they are, we don't know what the functions are. But by definition, a hormone is biologically active. Let's look at an example here. This is one that is frequently used as an example to illustrate this process. So here is a pro-hormone called POMC, propionmelanocortin, and the name kind of tells you some of the hormones that are packaged into this precursor. So this long pro-hormone includes several active hormones that must be proteolysis, proteases are going to chop up this pro-hormone into active components. Pro-POMC would include, for example, ACTH, which is going to drive release of adrenocortical hormones, originates in the pituitary. This precursor protein would also include species of melanocyte stimulating hormone that would alter pigmentation. Therefore, if a patient has a disorder, has hypoadrenalism or hyperadrenalism, you might anticipate changes in pigmentation would result because as a first approximation, they're going to be secreted together. Endorphins, we've talked about these being endogenous analgesics. They have some other functions too. Here's another type of MSH. This, as far as I know, is only present in humans. It's only humans. Endorphins and kephalins. So these are just some examples of the active biological compounds and hormones that would all be packaged together in one pro-hormone we're calling propionmelanocortin. And as we're going to find out, this would be, for example, present in the pituitary gland. All right, moving on to the next next group, we talked about the steroids, we talked about the synthesis and the general characteristics of peptides. There are also the remaining amines. Remember, we sort of, we're going to call these amines or amino acids in this context. The distinction is not terribly important. Problem is, this chemical class of amines includes the catecholamines and the thyroid hormones, and they operate rather differently. The catecholamines are all synthesized from tyrosine. They do not have the ability, as we know, to diffuse across the membrane. They bind to membrane receptors. They are typically unbound. In some cases, they can be loosely bound to chaperones in transit. And they have very short half-lives, very, very short, on the order of seconds to minutes. The thyroid hormone, on the other hand, while it does require tyrosine, it also requires iodine. That's why your Morton salt is iodinated. They do have the ability to cross the membrane, although we need to talk about this. This is going to require some talking because they are hydrophobic, but they don't really follow the usual rule you would expect of hydrophobic molecules. They bind intracellularly. Remember, they act like steroid hormones. They bind intracellular to act as transcription factors. They're going to be stored and released from the thyroid follicle. We'll talk a little bit about thyroid hormone synthesis. They do have to be bound in transit because they're insoluble, relatively insoluble in the blood, so they're going to be bound to. Remember, we looked at it earlier. What was the chaperone for thyroid hormone? We looked at it in that little. Yeah, TBG, thyroid binding globulin. That's the most common one. And they have a much longer half-life, largely because, once again, of this circulating chaperone. So here's a little table that summarizes the characteristics that we've been talking about. In this case, I've put the catecholamines and the thyroid in separate columns here. You pretty much need to know this table. So I think you already know this. I think most of this is pretty evident to you. We've talked about solubility. I don't think you'll see any surprise there. We've talked about the synthetic pathway. The peptides are the only one we really talked about in detail, but you should know that we'll explore this in more detail later, especially as we talk about the reproductive hormones, for example. Multiple enzymes are required in the synthesis of all of these. We'll also talk about a few of the enzymes involved in thyroid hormone synthesis in just a moment. Let's skip this one for a second. Protein bound just means do they need a chaperone or not? And so the steroids and the thyroids do. Again, there are some exceptions for some of the others, but this is the general principle. Pay attention to the half-life here. I do want you to be familiar, sort of place them in a hierarchy with respect to the relative half-life in the blood. And then make sure you know where the receptors are. Remember, steroid receptors can go either way. They usually bind intracellularly, but in some cases, especially estrogen receptors, aldosterone receptors, for example, the hormones bind at the cell membrane. And then we'll talk about the mechanisms in more detail, too, in just a moment. Now, with respect to this storage thing, it says the peptides can be stored, catecholamines can be stored, steroids cannot. Why is that? Why don't I have a check mark by the steroids for storage? We've actually talked about this before. Steroids, what are their chemical properties? Can they diffuse through membranes? So why would that present a problem for intracellular storage? How are you going to lock them up? Yeah. You can't confine them. They've got to be free. And so, as a consequence, we don't put a check mark here, but then that presents this. Why do I have a check mark under thyroid hormone? Because it's very similar. And the answer is the thyroid hormone is stored in a very special way. It's sort of stored in a precursor form. You have about 2 or 3 months of thyroid hormone chucked away in your throat there, but it's in a precursor form, and then it's only activated when needed and released. Yes?
Is that why you said that thyroid hormones can also pass the cell membrane? Because they're doing so in the precursor form?
As I said, we need to talk about that. They, I'm not sure what to tell you because the science, well, I can tell you what happens. I'm not sure I can explain it to you because I've never seen a very good explanation. What we're going to find is that although thyroid hormone, and by that I'm talking about the active thyroid hormone, remember that little diagram we saw, the T4 and the T3, although it has the ability to pass through the membrane, it's actually actively transported. So that's something it's in the notes, and we're going to talk about it again later, but it's something you definitely need to know because if your patient has a problem with the thyroid hormone transporter, they've got serious issues. And again, it doesn't follow from the chemical structure of the molecule. All right, so now we're going to turn our attention, and it's not necessarily clear from the change in the topic here. We're going to talk about what I'm calling endocrine signaling. But make sure you understand, make sure you, as we enter this topic, you understand we're only talking about the water-soluble hormones. Now we're talking about the amines and the peptides. And we've already talked about the importance of chemical structure. We know that the peptides and the amines must bind to membrane receptors. I'll sometimes say extracellular, but what I mean by that is membrane receptors. And so that presents a problem, and that means, once again, if the room is a cell here, as I'm sure all of you are increasingly understanding it to be, as in jail cell. So we have a cell here, and this will be won't be a voltage-gated channel this time. It will be my hormone receptor. And so that's the blood out there, and the water-soluble hormone is transiting in the blood back and forth, and from time to time, it just knocks at the door. But what I want you to understand is that's no big deal. That doesn't. In order for a hormone to exert or induce or produce a biological response, it has to cause something to change inside the cell, and simply knocking at the door of a receptor is not going to do it. So when we talk about endocrine signaling, we're talking about that sequence of steps that must occur between binding of the water-soluble hormone to the membrane receptor and then something occurring or changing inside the cell. So again, just remember we call that second messenger signaling, signal transduction. But that's what we're looking at in this context. We say that these hormones, and again I'm talking about the water-soluble hormones, bind receptors with high specificity. That's usually true. That's probably even more true than with neurotransmitters. High affinity, it means that there's a strong association between the receptor and the hormone. But relatively low capacity. And all that means is that you can see in the room here, we only have 2 receptors, and that's going to be true for a lot of cells. Cells are limited in the resources that they can devote to hormone receptors, and there are only going to be so many along the membrane, and therefore the ability of that cell to respond to a hormone is going to be very much determined by the receptor abundance, receptor density. We've already talked about how cells can change their sensitivity or the relative abundance of those receptors. So here's a pretty grim slide, I'll have to admit. But what I've done in a rather simplistic and probably inappropriate fashion is I've categorized the major endocrine signaling mechanisms, I've just thrown out a few of these, into 4 general categories here. And these are the ones you need to know. These are the ones, and by the way, you can see we've already talked about the steroids, so we don't need to worry about that. So there's really only 3. There's the adenylyl cyclase pathway, the phospholipase, or the PLC. The first one's also called the PKA. So the other one's also called PKA, number 1, protein kinase A. Number 2, the phospholipase C, or PLC pathway. And then the third one is used by insulin and a lot of the growth factors. We're just going to call it the tyrosine kinase pathway, or tyrosine kinase signaling. As I say, I've done away, I've dispensed with some of the less critical pathways. Now what you'll notice here in this table the PKA, remember that's number one. We're going to use this protein kinase A as interchangeably with the number one adenylyl cyclase or adenylyl cyclase pathway. So look at all the receptors. You don't need to memorize this right now. As we work through these, at some point, you're probably going to have to memorize a lot of these. But the glycoproteins, for example, we're going to find in the pituitary hormones. A lot of pituitary hormones, we're going to fall into this category. So they would bind to anterior pituitary, bind to PKA receptors. Beta adrenergic. Later we're going to talk about glucagon, important or kind of important in regulating metabolism. Parathyroid hormone utilizes PKA receptors when it acts to modify circulating calcium ion concentration in the blood. We just looked at ACTH. ACTH is a cleavage product of POMC that drives release of adrenocortical hormones such as cortisol. Some of these are less important, but one thing, I don't have it listed here, so maybe add to this list the following, because I left out probably the most important ones. I'd say at least half, if not, I have to think about it right now. But we'll just say many of the hypothalamic hormones, that you're going to memorize shortly, utilize the PLC receptors. So let me say that again because it's important and I'm not sure why I left it out. Many of the hypothalamic hormones, which we're going to call releasing factors or inhibiting factors, utilize PLC receptors. One of the serotonergic, again, alpha adrenergic, calcitonin is going to be important too. Well, kind of important in regulating calcium. And then some of the muscarinic receptors. So again, you don't have to memorize this list. I just want you to be aware that a lot of these physiologically important pathways depend upon one or the other of these signaling sequences. And this is a kind of simple-minded way of me just reminding you that, once again, if we're the biological cell and that's the blood out there, the hormone is the first messenger. And that's all a hormone is. It's just a messenger. It doesn't do. Don't tell me that a hormone regulates glucose directly or that it increases metabolism. Really, all it does is tell various structures in the cell to do what they would normally do on their own. So that's the first messenger. And then for simplicity, just so that we all are using the same sort of lexicon, the second messenger will be the molecule that carries that signal from the membrane to the interior of the cell. That isn't always the case, but I think that's a good basic definition. So first messenger is the hormone, the second messenger is whatever carries the message to the interior of the cell. All right. So when that second messenger leaves the membrane and does so with the intent, with the purpose of exciting some intracellular biological response, what types of response might ensue? And I've just listed some of these. And you have information to add to this list. For example, when we talked about smooth muscle. Is it possible for smooth muscle contraction to be excited by hormone? Yes. So muscle contraction can also be added to this list. But certainly altering enzyme activity. So in other words, one of the most common consequences of second messenger signaling is to spread of fire within the cell. When I say spread of fire, I'm talking about moving phosphates, PO4s, from one molecule to another. Phosphates are what biochemists call activation energy. When one molecule acquires a phosphate, i.e. ATP, it has the ability to do all sorts of miraculous energetic things that it previously could not do. So we can sort of liken that to charging the battery or spreading the fire. That's often the consequence of the second messenger pathway. Altering intracellular calcium levels. We saw that previously too. If we open up that SR in a cell, or even the ER, and sometimes even the nucleus and the Golgi hold calcium, I think you realize what can happen. Muscle contraction, neurotransmission, exocytosis, changing in the cell receptors, mobilization of transporters, all of these things can follow changes in calcium concentration. Why would opening and closing ion channels be useful?
hypopolarization.
Yeah, we're talking about, for example, we talked about in the eye. Remember what enables you to see? What is responsible for the amount of neurotransmitter released from your rods and cones? Remember, it was a transducin. The transducin is a G protein, right? And that G protein is just a second messenger. And then we've already talked, I'll give you one example, but there are many of these. I want you to understand also that peptides and amines, although they bind at the membrane, they can also have a genomic effect. It just takes a little longer. It's a little more complicated. It involves many more steps. But they too can act as transcription factors. All right, let's take a break then. Resume.